Greetings from the World Health Organization. I'm Soumya Swaminathan. I'm the Chief Scientist, and I'm delighted to speak to you today. I'd like to speak to you today about HIV, TB, and COVID, and how these three have intersected, and share some ideas and views with you about what we can do in the future to leverage some of the exciting new advances that we've seen this year. I'd like to run you through a little bit of the epidemiological situation very quickly on COVID, talk about health system challenges, and then talk more specifically about the impact of COVID on HIV and TB and on children, TB and children, because I think that's of special interest to this group. And then what can we learn from the COVID pandemic to change the way that we do things. Now, globally, the situation has been fluctuating. As you know, we've had months where the pandemic seems to, have come down over the summer last year, but then you see the waves that we've had, the second and third wave in many countries, starting from November, December, and January. This graph actually looks it's color-coded by WHO region. So the region of the Americas is coded in yellow and the European region in green. These are the two that have borne the brunt of the pandemic, particularly over the last several months. And then you have below that the Southeast Asian region, where the epidemic is now on the wane, and then African region and the Eastern Mediterranean region. Now, in the last week, we had about 360,000 new confirmed cases and just under 10,000 new deaths. This is down from about 3 or 4 weeks ago, when we had a peak of 600,000 new cases every week, almost 100,000 new cases a day globally, and about 16,000 deaths every day. So there has been a decline in the last 3 or 4 weeks, but again, it's too early to relax. And of course, cumulatively, we know that we have over 110 million cases. These are confirmed detected cases, and close to 2.5 million deaths. And you can see the countries that had the highest number of new cases, again, mostly in Europe and in the Americas. This graph is a way to look at it in a color-coded way. It's cases per 100,000, and again, you can see the impact has been in North and South America and Europe mostly, but also in the Southern African region. Again, we can see the deaths per 100,000 population. Again, the brunt has been in North and South America and in the European region. And there are many reasons. One of them, of course, is demography. More older people in the more developed world, and therefore they've had a higher share of the deaths as well. So again, epidemic curve by region is interesting. It's different in different regions, again, something for us to think about. But again, showing that you can go up and down and then go up again. So one needs to be really very careful and not conclude that the pandemic is on its way out. Health systems have been quite badly affected, both because of the health impact on health workers, the lack of supplies and equipment. We saw early in 2020, there was a complete breakdown of global supply chains, and simple things like gloves and masks and personal protective equipment were not available in many countries. Huge burden on healthcare workers, disproportionate impact both in terms of illness and in terms of just being overworked and stressed, and disruption of the health services. So again, we were caught unawares, couldn't balance the demands of responding to COVID-19 with all the other services that need to be delivered. So the WHO did a pulse survey in July last year to just look at how many countries actually had plans in place to continue to deliver essential health services and how those were impacted. And we have repeated that pulse survey now, so we should have the results in a couple of weeks. What we found was that a large number of countries reported partial disruption in at least 75% of their services, and this was much higher in low-income countries compared to the upper middle income or the high-income countries. But there was a huge impact globally. And looking at which essential health services, looking at the top 25, we see that majority of countries reported a breakdown in routine immunization, dental rehabilitation services, and then going to things like TB, malaria, but also non-communicable diseases, cancer care, palliative care, but also things like blood transfusion services and essential surgery. So we know from countries that have good data systems that the impact on excess mortality over and above COVID-19 deaths has been quite significant, and that's partly due to the impact on these essential health services. Again, something to think about for the future. When we think about resilient health systems, we can't have everything else collapsing as we are responding to an epidemic. So we need both the workforce and the other items in place in order to be able to cope up. Now, what about the impact on HIV? There were significant disruptions during the period last year when there were lockdowns in many countries because there were movement restrictions, people couldn't get to health centers, there were local stockouts, there was decrease in the use of health facilities because of fear. And at that time, we had warned that these disruptions could really impact the advances that have been made in HIV, particularly in mother-to-child prevention services. And again, we found that 85% of HIV programs reported some disruption to service delivery. This was moderate in 66% and very high in about 5%. This was data that was collected by the Global Fund. This happened, of course, as I mentioned, across many other essential services. There was modeling conducted by WHO with UNAIDS and the Stop TB Partnership, both on the impact on HIV as well as on TB, and it was found that you could have an additional 500,000 AIDS-related deaths within a year compared to 2018 because of the COVID pandemic and the impact that it's had through disruption of services, both in diagnosis of new cases as well as in the continuation of prevention and treatment programs. Similarly, when we look at TB, what happened was that there was a lot of reallocation of human, financial, and other resources from TB to COVID response. So many countries started using gene expert machines for COVID testing because if they didn't have RT-PCR labs available, this was easier. It was just a question of procuring the cartridges. Reassignment of staff. So many TB staff, are accustomed to doing outreach and follow-up, and so they were redeployed to attend to COVID. And then there was, of course, reallocation of budget. So we had, in the survey, documented how many countries had done each of these things. Of course, again, because of similarities in the symptoms, there was also the potential for confusion with the stigma and further hindering case finding. And again, the survey showed that in many countries there was reduced access to care, impacting the entire care cascade, starting with prevention, diagnosis, treatment, and so on. And again, a modeling that showed that if these disruptions were not quickly attended, you could result in an additional 500,000 or so TB-related deaths, particularly multi-drug resistant TB, which requires access to diagnosis and to the proper treatment. Again, looking at the period last year when the lockdowns were in place in many countries, you can see that in 4 of the high TB burden countries, there was a significant reduction, 50% or more reduction in TB case detection. And obviously these would now, may result in additional TB deaths, and that would only be recognized much later, perhaps during the course of this year. But what is more worrying really, looking at the longer term impacts, is that the economic impact of the pandemic is predicted to worsen both GDP per capita as well as undernutrition. We're already hearing about the number of people who have lost their livelihoods, who are being pushed into poverty. There are very fragile and vulnerable countries as well as communities who were just on the edge, and the pandemic has really pushed them over that edge. We know that TB incidence is inversely related to GDP per capita, you can see on this figure, and that it is directly related to the prevalence of undernutrition in the population. In fact, India, where I come from, the main driver of tuberculosis is undernutrition. It is not HIV, which it is in many parts of sub-Saharan Africa, and therefore any increase in undernutrition is going to result in an increase in TB incidence in the coming years. So we need to be prepared for that. What about the impact particularly on women and children? Now, the pandemic has had a disproportionate impact on women and children in many ways. Luckily, COVID-19 did not have as big a biological impact on children. They're young children less likely to get infected, and if they get infected, they're much, much less likely to become severely ill or die. Of course, you have the multi-system inflammatory syndrome. It's relatively rare, and it has been reported in many countries, similar to, it looks like Kawasaki or toxic shock syndrome. So that is a complication, but it's rare. The majority of children are asymptomatic or have very mild illness. Of course, again, if they have comorbidities like asthma or diabetes or chronic lung disease, they could get more severe illness. But again, the experience and the data from low-income countries where there are more risk factors for poor outcomes in childhood pneumonia, for example, like malnutrition, HIV, we really don't know. There haven't been enough studies done to document the interaction of those factors. As far as TB in children and COVID is concerned, again, there isn't a lot of data, but you could expect poorer treatment outcomes for many of the reasons that we discussed. It is likely also that you could have concurrent TB and COVID, particularly in the high TB burden countries. The clinical picture could overlap, so you might have confusion. The right diagnostics need to be done, and we know that in children, symptoms of TB, symptoms and signs can be quite non-specific. So wherever we need to make sure there's a family history taken, that there is a history of whether there has been failure to thrive, looking for extrapulmonary manifestations underlying HIV, making sure that a chest X-ray is done, and so on, because we know that child survival from TB really depends on early diagnosis, and particularly in the youngest children, where you have the possibilities of it progressing very quickly to TB meningitis or miliary TB. It's important to have that high index of suspicion. TB is still underdiagnosed and underreported in children. We estimate about 12% of the 10 million new cases every year are in children, and about 230,000 deaths that occur in children under 15 years. But many of these are missed, and the deaths are considered due to other causes. Now again, on the TB care cascade in children, respiratory infections, you can confuse it with other diseases. The lockdowns resulted in delays in TB diagnosis and treatment, just like in adults. There could have been increased household exposure to TB because everybody was locked indoors. But again, we don't know the impact of mask wearing. And if the mask wearing has actually resulted in a reduction in TB transmission or not, that would be an important thing to find out. Competing needs for diagnosis, complacency about the risk of severe COVID in children, and also disruptions in production and supply of child-friendly formulations. But I think more important, the impact on children has been the indirect impacts of reduced household income, increased poverty and food insecurity, missed vaccinations and health checkups, being out of school for almost the entire year, sometimes missing out on midday meals, increased domestic violence, increased risk of trafficking. All of those increase the vulnerability of children to diseases like TB and also HIV. I think a few of the lessons to be learned about the way we've done contact tracing. Many of the countries that were successful in controlling COVID were countries that are used to doing this for other infectious diseases like TB and HIV. They had more of a public health approach, and staff, community health workers and others on the ground who are used to visiting communities, going house to house, doing contact tracing. And so it was seen that in countries like Vietnam and Thailand and even countries like India, the contact tracing, the quarantining, the isolation and the follow-up of both people infected and exposed to COVID was actually much better than in many high-income countries where that public health infrastructure over the years has no longer been maintained at that at that level. We need to use more of digital tools and technologies, perhaps reduce the number of visits that people have to make, use the kind of mobile apps that were developed for tracking of COVID, try to provide the drugs in combination to people for longer periods of time and then keep them under follow-up through audio or video calls, use fully oral treatments for multi-drug resistant TB so that you reduce the contact with the health system, delivering medications at home, dispensing it to a caregiver instead of making the patient come. Many of things were actually done by countries during the last year. And then infection control in institutions. Again, there's been a new emphasis now on infection prevention and control because of COVID, on mask wearing, on hand washing, on disinfecting. And so some of these practices we need to continue because they could have really big impacts. You know, I remember when I was in India, there was so much of a reluctance to mask wearing. This is pre-COVID, of course, because it was thought that it would create stigma if you ask TB patients to wear masks. But if everybody in a medical facility is asked to wear a mask, then there's no longer any stigma. And now I think the whole world has got used to wearing masks. So maybe that's one good. We've seen influenza, for example, both in the southern hemisphere and the northern hemisphere. There's been no influenza at all this year. So the measures that we've taken have obviously controlled to a large extent the influenza epidemic that we would have every year. So let's see what impact it has on TB transmission. So we need to ensure that, you know, the focus on TB diagnosis, particularly for children, does not get left behind because of the focus on COVID. We may have to test children for either or both diseases, and in some cases the testing for both TB and COVID might be indicated if there is a clinical picture that's common to both, if there is exposure, simultaneous exposure to both, or if there's a risk factor for poor outcomes, like particularly I'm thinking about malnutrition or underlying HIV. Specimen collection in children has always been a challenge, whether it's a gastric aspirate or a bronchoalveolar lavage. These procedures do tend to generate aerosols and droplets. There are non-invasive specimens like stool, for example, that are low risk, easy to collect, but again, not yet widely used in many places for diagnostics. Finally, I'd like to say a little bit about what we have been doing for TB. The Stop TB department in WHO has created a compendium of publications and research projects related to TB and COVID-19. They are monitoring all the research findings to support national TB programs and also supporting efforts to compile individual patient-related data on people who've had both COVID and TB so that we can learn. I think it's important to do that kind of operational research. There's a digital library of publications, clinical trial information, particularly a lot of interest on BCG, as well as good practices on TB and COVID-19 that have all been put into this compendium. There's also an information note that was produced a few months ago on ensuring continuity of TB services during the COVID pandemic. We did this for many of the services, for immunization services, for antenatal services, and so on, how countries could actually ensure the continuity of services. It includes, of course, community engagement in the development of these solutions, but also the use of digital technologies, proactive planning, and leveraging the capacity of staff on the ground to do multiple things. So this is the operational planning during pandemic. I mentioned that there was a pulse survey that was done. There was also this document produced on how to maintain essential health services when you have this type of additional shock to the system. As far as BCG is concerned, there are trials going on, and I think we need to wait and see. There was a lot of excitement early on that BCG vaccination might have a role to play in preventing the either the incidence or the progression of COVID and there was ecological associations made between countries that had widespread BCG vaccination and the incidence of COVID and the incidence of death due to COVID. But whether there's a direct correlation or not is not known. We do know that BCG stimulates the innate immune response, at least for a few months. We know in children, for example, in infants, that there's a reduction in the incidence of sepsis, for example, in children who've been BCG vaccinated. We need to ensure that vaccine supplies are there for the children who need them around the world. And so we need to be evidence-based and data-based, obviously, wait for the clinical trials before we promote it. There have been many lessons learned, I think, including the use of rapid diagnostic platforms. The speed at which rapid diagnostic tests have been developed for COVID has been amazing, using new technologies like CRISPR-Cas9 technology, paper-based diagnostics, rapid antigen-based diagnostics. I think we really need to make a push for better and easier and affordable and scalable diagnostics that can be used at the community level to diagnose people with TB. Contact tracing, the whole concept of this gives us an opportunity to strengthen now both TB infection control and contact tracing. And as I mentioned, digital innovations, experience with multi-month dispensing, experience with telehealth and mobile health, where you could provide services from a distance and still be in touch with patients. All of those, I think, are important learnings. And then finally, I think we need to innovate our approach to R&D of therapeutics. And I just give you one example of a project that we have for children. It's called the GAP-F. It's about accelerating pediatric formulation development and uptake. It's coordinated by the WHO, but has many, many, many partners around the world. And really looking at, it was started because there was a huge gap in the availability of pediatric formulations for HIV, and then it expanded to TB and now to cancer. Starting with what you need, prioritize what you need by evaluating the landscape, have a business development plan and a product development cycle, look at the regulation that would be needed, working with developers, with manufacturers of these products all the time. And then once you have a product, how do you actually adopt, procure, and roll out uptake, and then monitor its uptake. So this has been very successful for pediatric HIV formulations, and I think we really need to do something like that, an end-to-end process when we have a gap, whether it's a new TB vaccine or whether it's new combination therapies for prevention of HIV and so on. And then other speakers have spoken about pregnant and lactating women, but it's clear that we need this group of people to be included in clinical trials, clinical studies. We have spoken about this in TB and HIV, but I think with COVID, again, there's been an emphasis on not leaving pregnant and lactating women behind. There are ethical issues which need to be considered, but they shouldn't be excluded from all research because of the vulnerability. So we believe that I think the pandemic has really brought to the fore also some of the inequities that we've seen within countries and between countries in the way that health services are delivered, including for TB and HIV, and communities that are often very vulnerable and which have got further alienated or made even more fragile by the circumstances. It's really important to have community engagement in the whole process. I think we've learned again that countries that were successful in controlling COVID had political leadership, belief in science and public health, community engagement, and clear, transparent communication. I think these have been the elements of success, and we should learn the lessons from this and use the same as we look towards ending the epidemics of HIV and TB. Thank you very much.
Hello, my name is Jayne Sutherland, and I'm a TB immunologist working at the Medical Research Council unit in the Gambia in West Africa. And today I'm going to speak to you about the development of novel TB diagnostic tests. So, diagnosis of TB. In 2019, 10 million people fell ill with TB, but only 7 million of these were detected and officially notified. So a study in India showed that there's significant gaps in when individuals have symptoms to when individuals actually get treated. So the first gap is not having access to a TB diagnostic test. Gap 2 is not getting diagnosed. Gap 3 is not getting registered for treatment even when diagnosed. Gap 4 is not achieving treatment success. And Gap 5 is experiencing post-treatment TB recurrence or death. So diagnostic gaps are missing 3 million people per year. And this is particularly difficult for diagnosis of TB in children. So in 2019, 1.1 million children were diagnosed with TB, but this is likely an underestimate due to diagnostic difficulties, including difficulties in sputum production, the fact that if they do produce sputum, it's often poor subacillary, and that symptoms can be scant or non-specific. So we need effective diagnostic tests that can be performed on easily accessible samples. So for World TB Day this year, the theme is the clock is ticking. So we know that every year, millions of TB infections go unidentified, preventing patients from accessing treatment and allowing the disease to spread. So diagnostics are key to detect and stop the spread of TB. So there's several different diagnostic test needs, and this is to do with the TB spectrum, which is quite complex. So we need tests that can detect infection. So these are the next generation UCRU tests or interferon gamma release assays. We need tests that will detect incipient TB. So these are prognostic tests for those people at risk of development of active disease. We need tests for clinical disease. So these are triage or diagnostic tests. And we need tests that will determine whether someone is going to have successful treatment or is likely to relapse. So these are treatment response and relapse tests. So the two I will focus on today are tests for incipient TB and tests for clinical disease. So just to illustrate the difficulties with TB diagnosis in resource-constrained settings. So this is the TB diagnostic pyramid. And we see the percent of patients over here. So the majority of patients are accessing rural clinics with limited infrastructure, so level 3 clinics. The diagnostic tests needed here are point of care tests. Very few patients are able to actually access diagnostic centers that have the best diagnostic tests. So there's currently no point-of-care test for use in basic health clinics, which see the majority of all TB cases. And without diagnosis, transmission rates occur at approximately 15 contacts per patient per year. So tuberculosis elimination requires development of fast and affordable diagnostic tests. There are two areas here. So there's a target product profiles is what the WHO have defined for the specificity and sensitivity of tests that need to reach these levels to be used in clinical diagnosis. So the first one, target product profile 1, is for a rapid biomarker-based, non-sputum based test for TB, which I'm not going to talk about today. Target product profile 2 is for a triage test, which is a rule-out test for TB. So screening with this test can then determine whether someone is more likely to develop, is more likely to have TB, and therefore they require further screening. And this will significantly reduce the cost of laboratory tests, but they require about 90% sensitivity and 70% specificity as a minimal criteria. So the diagnostic vision for FINDS, a FIND being the Foundation for Innovative New Diagnostics, who help in the rollout of accessible and affordable diagnostic tests. So it's the first point of contact, which is level 0, level 1. So a triage test and highly sensitive detection at level 1. So whether that's a lateral flow test, which I'll go into detail in a second. Next stage would be dedicated unit level 1 or level 2. So this would be TB confirmation with rapid and integrated drug susceptibility testing for critical drugs to drive regimen decisions. And then we would progress through to reference level or level 3 tests, which are the comprehensive drug susceptibility testing, to cover the extended portfolio of old and new drugs. So this requires extensive infrastructure, power supply, all sorts of things, and they're obviously expensive. So current diagnostic tests are based on detection of the pathogen in the sputum. So this uses either acid-fast bacilli detection by microscopy, which is in worldwide use for detection of pulmonary TB. However, sensitivity can be as low as 50%, and this is particularly so in HIV-positive patients and children. Sputum culture is the gold standard. However, it's prone to contamination, can be very time-consuming, and it's very expensive. And I haven't put here, it also requires extensive infrastructure. So the Gene Expert test has been revolutionary in terms of diagnostics. It takes about 2 hours to perform. It's based on real-time PCR detection of the pathogen directly from the sputum, and it's fast and accurate. However, it is expensive and requires a constant power source. So current diagnostic tests are non-sputum based. So these can be based on host proteins, host RNA, cell stimulation, pathogen proteins, or antibodies. So tests such as the lateral flow test can detect host proteins in accessible samples such as serum, and this can be used as a potential triage test to rule out TB. Quantum Diagnostics have developed a handheld test based on detection of host transcripts in blood, and this has potential for use as a prognostic test for risk of developing active TB in exposed contacts. There's also Cepheid, who have developed the Gene Expert test, have also developed a cartridge which is termed the 3-gene host response cartridge. So it's detecting host transcripts, laboratory-based, for diagnosis of active pulmonary TB. Then we have the urinary LAM tests, which are currently indicated for use in HIV-positive individuals only, and breath tests and skin patches. However, all of these tests are still in development and then require multi-country head-to-head comparison in different patient groups, genetic backgrounds, risk factors, and epidemiology. So this is where our consortium comes in. Our consortium is called the END TB Consortium, and it's funded by NIH. So the aim is to perform head-to-head evaluation of prognostic and diagnostic tests for TB at different levels of the health system. So the sites include South Africa, Uganda, Gambia, and Vietnam. And participants include symptomatic adults, symptomatic children, and exposed contacts. So the tests that we're going to look at. So first aim is to look at adults and children with presumptive TB, and second aim is prediction of incident TB in exposed contacts, but all will use the same tests. So we'll look at the gold standard tests, which are symptom screen, clinical exam, chest x-ray, and sputum smear. And for microbiology, the Expert Ultra and the midget tests at regional labs. So people will present to the primary healthcare facility with symptoms suggestive of TB, but without prior microbiology confirmation. And this way we can test our research tests prior to knowing the final status. So the biomarker tests we'll look at are multi-biomarker test, a Cepheid 3 gene, and a urinary LAM test. We'll also be performing at central laboratories biomarker testing using mRNA signatures by Fluidigm and host signatures by Luminex. So just to describe what the multi-biomarker test is, so MBT. So this was developed in several different stages, and the first stage was biomarker discovery using serum samples collected during the African European TB Consortium, which was EDCTP funded. So Luminex analysis was performed of serum, and 7 markers were identified from approximately 800 participants, both HIV positive and HIV negative. And this showed 94% sensitivity and 73% specificity for TB compared to other respiratory diseases, which was shown to meet the WHO target product profile for a triage test. The next stage was to translate these Luminex markers onto lateral flow strips. So we used singleplex strips during a second consortium called Screen TB. And this looked at CRP, SAA, IP10, ferritin, ApoA1, and IL-6. So these were put onto basic singleplex MBT strips. The next stage, which is the current trial we're doing, is to use multiplex strips using fingerstick blood and see if we can evaluate these tests using point of care. So this is called triage TB and also the index TB. So we're looking at 3 markers, CRP, IP10, and SAA, and we're looking at fingerstick blood compared to serum. So this is just to go into a little bit more detail of the MBT test development. So as I mentioned, this was firstly a singleplex development. So we have a sample pad, which is nitrocellulose, and an absorbent pad, and we have a test and a control strips here. And you can see the strips standing up here. So they're put into wells, and then the lateral flow, they'll flow up there overnight, and then we can run them on a reader and look at the progression. We then went on to multiplex strips. So this has 6 markers on the one strip. Again, it's lateral flow, and we're able to, the lateral flow will flow overnight, and then we're able to detect on our reader. And the next stage, which is the current stage of our test development, is point of care. So adding the blood directly onto a cartridge and looking at the 3 biomarkers. So just to give you some information on the data so far, so prospective evaluation of fingerstick blood in the Gambia using a multi-biomarker test gave an AUC of 0.89 for detection of TB amongst all respiratory diseases. However, this was quite low participant numbers, hence why we need to do it in a larger field site study. So this will be evaluated in our multi-site study as part of NTB. So the next test I want to talk about are Cepheid tests. So we obviously use GeneXpert Ultra for MTB detection. So that's our standard test for screening of participants for TB. We're also performing COVID-19 detection on all our participants using the SARS-CoV-2 cartridges that have been developed by Cepheid. And at the same time, we're doing a host 3 gene, which is a fingerstick blood screening test for TB. And this is all on the one machine. Obviously requires more infrastructure than a lateral flow point of care test. So the Cepheid 3 gene was developed, so the 3 gene signature was developed by Sweeney et al., who showed that three genes could discriminate between TB and other diseases. So Cepheid developed these into an expert MTB host response prototype that quantifies relative mRNA levels of Sweeney 3 gene signature in a patient whole blood sample. So it's intended for direct analysis of fingerprint blood, but can also be used retrospectively with blood samples that have been preserved in PAX gene buffer. So NTB will evaluate this test using fingerstick blood in a prospective cohort of adults, children, and exposed contacts. And you can see here some results. So we get CT values for the 3 target genes, which are then put into an algorithm to determine whether it's TB or not TB. We were also performing a high sensitivity urinary lamb test in our study. So the current urinary lamb tests are based on detection of lipoaberramannan which is a component of MTB and is excreted in urine. Currently, urinary lamb tests such as ALEA or Fujilam, these are the 2 tests here, are recommended for use in HIV positive individuals, and they have low sensitivity in HIV negative individuals. But the next generation of urinary lamb are using generation of high affinity antibodies. So for index, we will use Mologic painted technology. In addition, there can be pre-concentration of the lamb in the urine and also analysis of sputum lamb for diagnosis and treatment response. Other tests that are in development include artificial intelligence for reading of chest x-rays, and this is quite an exciting area of research. So this shows the CAD4 TB heat map, which can read x-rays and detect whether someone has TB or not. So CAD can be used for TB detection for research, ideally following a protocol that contributes to the required evidence base for guideline development. QXR can detect and localize multiple findings in a chest x-ray, including detection of multiple findings with the lungs, pleura, heart, bones, and the diaphragm. It remains to be seen how useful this is in a point-of-care setting. So I next want to talk about detection of incipient TB, so risk of progression to active TB disease. So throughout the world, 2 billion people are latently infected, and therefore there is a need for targeted preventative therapies. Current products such as the UCRU and the TST, tuberculosis skin test, result in about 2 to 3% positive predictive value for detecting at-risk individuals, which is far too low. We have worked on developing a 16 gene test, which has a positive predictive value of 7%. So it's about 3-fold higher for detecting incipient TB. So how this was developed, we looked at exposed contacts followed for 2 years to determine incident TB. And then we're able to determine a 16 gene signature of risk with greatest accuracy. And this was shown to have greatest accuracy 6 months prior to progression. So it's likely detecting subclinical disease. And following this, we were able to reduce the signature from 16 to 6 genes while still retaining prognostic accuracy. So risk 6 has about 88% prognostic accuracy compared to 82% for the 16 gene. So the importance of a targeted therapeutic approach to identify those most at risk of progression will enable us to reduce unnecessary treatment, prevent subclinical disease progression, and hopefully this can occur with a handheld RNA signature device developed by Quantum Diagnostics. So the tests we're going to be doing, again, are point-of-care tests that we're using for our symptomatic individuals. So we'll compare the Cepheid 3 gene and the Quantum Diagnostic test. And the Quantum Diagnostic test is based on our published 6-gene signature. We'll recruit 2,000 exposed household contacts at all, combined at all study sites. And these will be followed for 12 months to determine incident TB. Other point-of-care tests will also be evaluated, including our multi-biomarker test and our urinary lamb test. We'll also collect samples for more specific laboratory evaluation. So we'll collect PAX gene, whole blood RNA from all participants, so we can look at multiple signatures which are already discovered, but also discover new signatures. And this will be performed using Fluidigm high throughput qPCR. We will also look at proteins or serum signatures using Luminex. And one final aspect I want to discuss is the importance of COVID-19 detection. So we know that COVID can present as asymptomatic or with similar symptoms to TB. So since TB and COVID are respiratory infections, COVID may worsen TB outcome, worsen lung damage, and or increase susceptibility to TB. And COVID may also impact on the accuracy of both current and novel TB diagnostic tests. So all participants in our study will be evaluated for SARS-CoV-2, regardless of symptoms, using PCR or the express cartridges, and also using serological assays. So we'll perform our final analysis to assess prior or current COVID-19 infection. So just to conclude, we have several novel diagnostic tests for different levels of the health system which are in development. Head-to-head comparison is occurring in different consortia. So these include hard to diagnose individuals such as children and patients with extrapulmonary TB, those with high risk such as diabetes mellitus, HIV, and malnourished individuals, and also exposed contacts to determine prognostic test accuracy. It requires high level and sustained funding. So the TB targets for 2022 were an increase in global investment for TB R&D to US $2 billion annually. And also but there's definitely been an impact of COVID on resources for TB. So to end, World TB Day, March 24th, 2021, the theme is "The clock is ticking to reach these targets." And just to acknowledge our funders and our collaborators. Thank you very much.
Dear friends, dear colleagues, I'm very pleased to join you on this World TB Day event hosted by the Oxford University Research Unit in Ho Chi Minh City. I wish I was there with you in person. In the absence of that, I'm most grateful to Professor Thwaites for kindly inviting me to present at the event today. I will be presenting the results of a phase 3 clinical trial that was just completed. I wish you all a successful World TB Day event. My name is Payam. I'm a professor of pulmonary and critical care medicine at the University of California, San Francisco, and I'm going to be presenting on behalf of my protocol co-chairs and protocol team, as well as the many others who have contributed to making this trial a success. The study 31 was conducted by the Centers for Disease Control and NIH, specifically the TBTC and ACTG. And as many of you may know, Vietnam, through the partnership with the Vietnam National TB Program and Professor Duong at our clinical trial unit in Hanoi, contributed to this study as well. By way of background, the length of time for treatment has been a long-standing public health goal. Shorter regimens will cure patients faster and have the potential to reduce treatment costs, improve patient quality of life, and increase completion of therapy, perhaps other benefits. The key study question was whether high-dose daily rifapentine, with or without moxifloxacin, would allow treatment shortening to 4 months for drug-susceptible TB. The trial had 3 arms with randomization 1:1:1 into control or one of 2 experimental or investigational regimens. The control arm was a standard 6-month regimen of daily treatment with isoniazid, ethambutol, and pyrazinamide. The rifapentine-only arm was a 4-month with a single substitution of rifapentine for rifampicin. The rifapentine moxifloxacin arm shown here was a rifapentine-containing regimen. In addition to this, substituted moxifloxacin for ethambutol, and then continues the ethambutol during the continuation phase. Both investigational regimens were 17 weeks in duration. All treatment was administered daily, 7 days a week. 5 of 7 doses were required to be given by directly observed therapy. Rifapentine was at a flat dose of 1,200 milligrams, regardless of participant weight. Moxifloxacin similarly was given at a flat dose of 400 milligrams. Because of differential food effect on these 2 key drugs, rifampin and rifapentine, rifampicin drug exposures are enhanced when taken on an empty stomach. Rifapentine are drug exposures enhanced when there's some food in the stomach. We were unable to blind the study, so in accordance with discussions with the Food and Drug Administration, open label design was recommended and pursued for this trial. The primary efficacy endpoint in consultation with the US FDA was an outcome at 12 months post-randomization. However, we will have follow-up through to 18 months post-randomization, which will generate additional reports in the future. Today, I'm presenting on the 12-month efficacy endpoint data. The eligibility criteria included positive sputum smears or a medium or high result on the gene expert. And one of the strengths of study 31 was the inclusion of adolescents aged 12 years or older. If HIV positive patients were identified, we permitted a CD4 count as low as 100. Insofar as exclusions, they were standard. Any prior anti-TB treatment of more than 5 days within the past 6 months was an exclusion. We excluded participants who had extrapulmonary TB that was particularly complex, such as CNS, bones, joints, miliary, or pericardial TB, and weight lower than 40 kilograms was excluded. The primary outcome of study 31 is shown here: TB disease-free survival at 12 months after study treatment assignment. Each eligible participant was classified into one of 3 outcome categories: cure or favorable, absence of cure or unfavorable, and then not accessible, shown here. I will show you later how each of these categories had the participant makeup of these different categories. After each participant had a primary outcome into one of these three classifications, two co-primary analysis populations were formed, which were shown here, the microbiologically and the accessible population. The difference between these two populations is that the not accessible participants in this yellow box were classified automatically as unfavorable in the microbiologically eligible analysis population, whereas they were excluded in the analyses of the accessible analysis population. So they give you two different viewpoints of the data, and I'll go over those later. It was conducted with broad geographic diversity, as you can see here, 34 clinical research sites were involved, 13 countries, 4 continents. TBTC sites sponsored by the Centers for Disease Control are shown in green, including the site in Hanoi here, which we at the VNTP UCSF research collaboration conducted the trial. And then the red show the ACTG sites funded by the NIH. Broadly, you can see a very wide geographic representation. This figure shows the recruitments by country, again demonstrating the diversity. The top enrolling country was South Africa, with multis in South Africa participating. And this was followed by Uganda, then Haiti, and Vietnam, as the site in Hanoi was fourth, and the rest of the sites are shown here in terms of their contributions. This figure shows the consort diagram of the clinical trial, and what you can see is that over 5,000 patients were screened, and the meeting eligibility for randomization, 2,500 underwent randomization, allocated 1:1:1 with equal distribution across the 3 arms. Next, from this intention to treat population, we exclude patients who were randomized but then found to have baseline drug resistance to rifampicin or isoniazid. They were excluded, and if any did not have baseline positive cultures were excluded. And that leaves us with the microbiome-eligible groups shown here. That's around 760 to 790 participants. Then we further have exclusions shown here, where participants were lost to follow-up after completing treatment, even if their last culture was negative, or if they stopped therapy during these are the sort of not accessible population, and that results then with that exclusion we're left with about 720 to 750 participants in the accessible group. And I will show you again that we do analyses in both the microbiologically eligible group and the accessible group to really evaluate the performance of the regimens. This table shows the baseline characteristics of the microbiologically eligible population. The total in this population is again shown here, 2,300 patients, thereabouts. 70% were male. This is consistent with other contemporary clinical trials. The age was a median of 31 and balanced across groups. The race of participants was also diverse. The majority were Black or African American, given the countries that contributed the most to this enrollment, not surprising, but certainly a diversity of races included. There were about 8% HIV infected population. This corresponds to the global epidemiology of TB-HIV. And in terms of severity of disease, I would sort of say that this was a relatively severe population with a severe burden of disease, with 72% of them having cavitary disease on baseline chest X-ray, and showing malnourishment across the population, with a median weight of 53 kilograms. This slide shows you the results. These are the primary efficacy analysis results. And I am just thrilled to show you that the study had a non-inferior regimen. But before I get there, let me first point out that the study had a non-inferiority margin of 6.6%. And so just if I could take a moment to explain that, that means in order to demonstrate non-inferiority, the upper bound of the 95% confidence interval for the difference in proportions of unfavorable outcomes between investigational and control regimen should be below 6.6%. Further, non-inferiority would have to be demonstrated in both the co-primary analysis populations, that means both the assessable population here as well as the microbiologically population shown here. The upper graph shows the results of the rifapentine moxifloxacin regimen. You can see that for both co-primary analysis populations, non-inferiority was met. Note that the upper bound of the 95% confidence interval is well within the margin. So here's your 6.6% margin. These are the upper bounds, and you can see they're well within that margin for both populations analyzed, the assessable and microbiologically eligible. Solid circles versus open circles just illustrate that findings are consistent when adjusted for HIV and presence of cavities on baseline chest X-ray or when they're unadjusted. And so this is reassuring. On the lower graph, you can see the comparison of the rifapentine arm versus control. And here you can see that the upper bound of the confidence interval for differences in the proportions of unfavorable outcomes between rifapentine only regimen and the control was above or at best at 6.6% in both the analysis populations, both the assessable and the microbiologically eligible. Therefore, the rifapentine does not meet non-inferiority criteria for efficacy. Now I'm going to show you details about the three classifications of favorable, unfavorable, and non-assessable. So first, on an outcome status of favorable, this slide shows that in the total of the total analysis population and the assessable analysis population of 2,234, 88.1% of the total population was favorable. You can see 90.4% control arm and 88.4% in the rifapentine moxifloxacin arm, lower for the rifapentine arm. Another feature of this that I think is worth commenting on is that the absolute majority of those who had a favorable outcome had microbiologic confirmation with being confirmed culture negative at 12 months, which is quite reassuring and a measure of robustness of the trial. In fact, those who were seen at 12 months but had no sputum produced or culture was contaminated or unavailable was very small, only 34 participants. The other thing I'll point out here, just for point of reference, and I'll come back to this, is that even the regimen of a single substitution for rifapentine for rifampicin, nearly 86% cure rate is a significant improvement over the prior single substitution fluoroquinolone trials with gatifloxacin and moxifloxacin that did not have an optimized rifamycin in its backbone. So I think it's an interesting enhancement of cure with this strategy of optimizing rifamycins, and clearly with the moxifloxacin added even further enhanced. Now I'm going to show you the outcome status of unfavorable, those who are in the absence of cure classification. And what I can show you here is that overall, 11.9% of participants experienced an outcome here, with 9.6% in the control arm, 14.2% in the rifapentine arm, and 11.6% in the rifapentine moxifloxacin arm. We further then split up the unfavorable outcomes by TB-related outcomes versus non-TB-related outcomes. And the majority of participants with TB-related outcomes, the higher, this section here, defined by having 2 positive cultures after week 17. So that's this category here. So the total was 108 out of the 144 were culture confirmed. Now, there are other categories in this TB-related outcomes category, including loss to follow-up, with the last available culture being positive for TB, a change of treatment due to clinical recurrence without positivity or microbiology confirmation, or the development of extrapulmonary TB. Lower down here, I'm showing you the not TB-related outcomes of the total unfavorables. And these included with withdrawal of informed consent, treatment changes due to adverse event, death during treatment, moving away, poor adherence, loss to follow-up. Losses to follow-up were classified as unable or not accessible. That is, we put so much attention on improving retention for the analyses as robust and reliable as possible. You may note that the almost half of the unfavorable outcomes were actually not TB related. So the total population, 11.9%, 5.4% of the total 11.9% was not TB related. Now, finally, the not accessible group. This is a summary of why participants were classified as non-accessible. The total was 109 participants with this outcome. The main reason was loss to follow-up, with the last positive culture being negative for TB. Remember, if they were positive for TB, they would have been an unfavorization. In this case, their last culture was negative for TB, but they were not at 12 months. And so they are deemed as not accessible and allow us, then that will impact the analysis, as I showed you earlier, in the microbiologically eligible populations, wherein these participants would be defined as unfavorable, and in the accessible population, they would be excluded. Additional reasons included withdrawals from treatment due to pregnancy, or violent and accidental death during treatment, and exogenous reinfection as established by whole genome sequencing and retreatment for that. One of the really remarkable outcomes of our analyses was the consistency in which the regimen's performance was demonstrated. In particular, here I'm showing you the rifapentine moxifloxacin regimen that met non-inferiority criteria for efficacy. This regimen, in fact, continued to perform consistently across 14 sensitivity analyses that were pre-specified in the statistical analysis plan that effectively definitions of outcomes, varying definitions that took into consideration culture status, longitudinal culture status, and across the board, this regimen was non-inferior for all of those. Conversely, rifapentine substitution arm that did not have moxifloxacin did not meet non-inferiority criteria for efficacy in any of the sensitivity analyses, again, showing the upper bound of the 95% confidence in the margin of inferiority. Now, I think this is a really nice slide to think about the relationship risk of bias and different analysis populations in clinical trials. And I thank my colleague and friend, Patrick Phillips, for developing this slide. I think what's nice to look at here is the relationship between these different populations, study populations that I've walked you through, from the intention to treat to the microbiologically eligible, the accessible, TB-related unfavorable, per protocol is 75%, per protocol 95%, and how this sort of on this spectrum of populations, with this being the highest number of participants included, the most inclusive, and this one being the most restricted, how this interplays with bias. So for the intention population, which is the most inclusive, the biases there are introduced, classifying non-TB events as unfavorable, regardless of whether they were related to the therapy or not. And so that adds an element of bias there. On the converse side here, the per protocol, where these are the participants, and there are fewer of them, who followed the protocol with absolute exactness and adhered fully to all doses, never had gaps in care. One would think that that's invaluable and represents the performance of the regimen. However, there's biases introduced there through the fact that participants are excluded based on post-randomization data, and this departs from randomized comparisons. And so what is nice about the two co-primary populations selected for analysis for this study, the microbiologic eligible and the accessible, is that there's somewhere in between on this spectrum. And for us to be convinced, for all of us to be convinced that these results are consistent and robust, we need to show that the non-inferiority was present in both co-primary analyses, these two populations, and most secondary and sensitivity analyses. And then when we have that, then we can have some confidence that the risk of bias is minimal, and we can be more certain of the consistency of results. So I'm going to show you how those populations I just showed you on that figure play out for the two regimens. So first, I'm going to show you the rifapentine moxifloxacin arm, and then rank ordering the largest pool of participants, which is the intention to treat, through to the smallest pool of study participants characterized as the per protocol 95%. And you can see the denominators here are reducing. And what you find is that the point estimates and the 95% upper bound of the 95% confidence intervals all consistently well within the 6.6 margin of non-inferiority. This consistency gives us this confidence that the risk of bias is low and that our findings are robust. In the lower graph, I'm showing you the rifapentine regimen without moxifloxacin, and again, intention to treat stepwise down within the decreasing participant pools. But what you can see is that the two co-primary populations, microbiologically eligible and accessible, I told you before, did not meet non-inferiority, with these upper bounds being above the margin. And as you move actually further towards the so-called protocol correct population, those who medications with absolute, with exactness and followed all the steps, which you would think would be the most reflective of the regimen's true effectiveness, did not improve, the kind of performance of the regimen. In fact, the non-inferiority was even more evident in that regard. So, I think these are interesting comparisons, these different populations, but for the successful regimen, the consistency is reassuring. The other thing that's very, very interesting to me, and I share for you for your consideration, is that if I bring your attention to this sort of microbiologically eligible population, this 0.7% unfavorable event rate in this rifapentine arm, the addition of moxifloxacin appears to enhance the cure by 2%, 2.2% to 15% here. But when you look this difference in the per protocol 75 population shown here and 95 shown here, from the rifapentine only regimen versus the rifapentine moxi regimen, the difference is actually closer to 4 to 5 in terms of the efficacy. And I think just further indicates the importance of including moxifloxacin in the 4-month regimen. So how does this rifapentine regimen in study 31 compare to other 4-month drug susceptible regimens evaluated in recent clinical trials? In this slide, I show you the regimens studied in recent, over the past decade and a half in order of increasing difference, the proportion unfavorable between investigational and control arms. And the rifapentine moxifloxacin arm is indeed the most potent regimen, 4-month regimen studied to date, and meets non-inferiority. It's the only one that has met non-inferiority. Interestingly, the rifapentine regimen in study 31, whereas it did not meet non-inferiority, it is still the second most potent and effective 4-month regimen studied to date. And I think that's an interesting discovery as it pertains to the initial hypothesis of optimizing for treatment shortening. Following that, the Oflatub regimen, published in 2013 with gatifloxacin and standard dose rifampicin, is the next most potent, not meeting non-inferiority, obviously, and none of these do after that. The 2 Remox regimens are shown. One of the stand regimens of pertominate at 200 milligrams with moxetinamide is next. The rifaquin 4-month regimen follows, and then finally the stand pertominate at 100 milligrams and moxie and pyrazimide is shown at the bottom. But I think broadly speaking, these 2 regimens in study 31 are the most and second most potent regimens ever evaluated to date. Now I'm going to shift to safety results. Very important as you enhance, optimize the rifamycin exposure, as well as add moxifloxacin to look for any signals of safety or tolerability concerns. So here are the primary and secondary safety outcomes. The primary safety outcome was the any grade 3 to 5 adverse event. And what I'm showing you here is the control, rifapentine and rifapentine moxifloxacin arms, shown in blue, red, and green respectively. And you can see that the rifapentine moxifloxacin arm is comparable to the control regimen in terms of grade 3 to 5 adverse events. The rifapentine single substitution arm was better tolerated than both the rifapentine arm and rifapentine moxi arm and the control, which is interesting. In secondary safety outcomes, I would like to show you that rifapentine only arm did had fewer adverse events and better tolerability overall as compared to the other 2 regimens. And other than the treatment-related grade 3 to 5 adverse event categorization, where numerically there was a higher proportion of grade 3 to 5 adverse events reported for the rifapentine moxi arm, the tolerability, serious adverse events, death were lower in each of those for the successful 4-month regimen. Now, I think this deserves some more investigation. I'll show you some of the details of events, but I remind you that it was an open label, unblinded study where the principal investigator at the site did treatment relatedness. And I think that's an important thing to remember when looking at any analysis of safety when there's an open label study. And I think that looking with a little bit more granularity at the actual grade 3 or higher adverse events is helpful. And so here I'm showing you any AEs grade 3 or higher during treatment and up to 14 days after the last dose taken by the MEDRA system organ class, which is standardization of adverse events. And overall, you can see that the most common adverse event category was blood and lymphatic system disorders. This was primarily followed by hepatobiliary disorders with 3.6% of the population experiencing that. And notably, no significant difference between the arms as it pertained to either of these adverse event categories. I would draw your attention to the 3 arrows here, highlighted in part, well, in particular because of interest around fluoroquinolones, the inclusion of fluoroquinolones in this regimen, amoxifloxacin inclusion, and whether nervous system disorders, musculoskeletal connective tissue disorders would have been an issue. And in fact, there were very infrequent events and balanced. They were balanced across all arms in terms of these categories. You can see that these are very small numbers, obviously, but there was no signals of concern. Cardiac disorders were 3 total. All 3 were in the rifapentine moxifloxacin arm. Of the 3, 1 was a participant who experienced a QTc prolongation to 460 milliseconds and was discontinued from treatment after 3 doses of study drug. This was the only case of QTc prolongation out of the 2,500 participants that received treatment. Although it didn't reach 500, the customary threshold, it was elevated, and the principal investigator determined it's best to discontinue treatment. The other 2 of these 3 were congested air, and both were reported related to the study treatment based on patient history and as determined by the principal investigator. In regard to all-cause mortality and deaths during treatment and follow-up, overall, there were 36 participants who died during treatment or follow-up, and mortality was similar across all the arms, 1.4% in the control, 1.3% in rifapentine, 1.5% in rifafloxacin. And in the control arm, there was actually the highest proportion of TB-related deaths, whereas the proportions were similar at 0.5% and 0.4% in the 2 investigational arms shown here. And finally, we took a closer look at liver enzyme abnormalities during the study treatment and up to 14 days after the last study dose. We used indicators that have been recommended by the US FDA with guidance for industry. And we looked at this in 3 main ways. First, we looked at the proportion of participants with any transaminase value that is 3 or more times above the upper limit of normal, shown here in the light, the white row. And you can see that there was no difference between the control arms and the 2 investigational arms in relation to this benchmark. Next, we looked at transaminase thresholds of 5 times, 10 times, and 20 over the upper limit of normal. And again, you see that there were no differences across the arms. In fact, again, numerically, perhaps fewer were events in the investigational arms as compared to the control standard regimen. And then lastly, we looked at combining transaminase elevations here with total bilirubin increases. And there was a trend, a very slight trend across these 3 arms. You can see 0.9, 1.0, 1.2 in the investigational regimens. However, in the rifapentine arms, these events were mainly low-level bilirubin elevations and mild to modest transaminase elevations, and none were suggestive of severe hepatocellular injury or hyperbilirubinemia on that basis. So reassuring overall in regard to safety in the context of optimized doses of rifamycins, as well as the addition of fluoroquinolone, in this case moxifloxacin. So the study 31 A5349 conclusions, in summary, are that the 3rd regimen studied, this 2 HPZM, 2 HPM, met non-inferiority criteria for efficacy. And importantly, that non-inferiority was consistent in all primary and secondary analysis populations, all sensitivity analyses, all subgroup analyses. And I didn't show you the data, but this regimen met non-inferiority and was consistent in HIV, infected, adolescents, and diabetics, key systems of interest. Regimen 2, on the other hand, the rifapentine regimen alone did not meet non-inferiority criteria in any of the primary or secondary analyses, despite being the second most potent 4-month regimen that's been studied in contemporary trials to date. Reassuringly, high-dose rifapentine regimens were safe and and usually better tolerated than the control arm. And I think overall, the combination, these results, I think, show that this new 4-month regimen is the first successful, safe, and well-tolerated short treatment regimen for drug-susceptible TB disease identified in almost 40 years and is to be celebrated. I close by acknowledging the leadership and my protocol co-chairs of one, as well as the protocol team who are engaged on designing this trial that, frankly, design discussions first sort of go back to 2011, 2010 even. And so it's been a long journey, and I acknowledge them. I want to acknowledge several representatives from Vietnam, Professor Duong, and Dr. Ha, shown here. And then I would also like to acknowledge the CDC and NIH for the operation on this trial, their funding, joint funding of it. It's really a mammoth undertaking, expert oversight of data and coordination provided by the CDC, drug supply and PK testing of TB drug aided by Sanofi. We are grateful to the Data Safety Monitoring Board of the TBTC for their oversight and monitoring. The 34 clinical trial sites in 4 continents, the 2,500 participants, their families and friends are all part of this journey. Lastly, I want to acknowledge the importance of including community representation, which we received from these 2 groups, and the importance of that in protocol design, in encouraging us to be as inclusive as possible in the eligibility criteria of the trial, so that the results could be translated and adopted rapidly by policy decision makers. With that, I thank you very much. I wish you all a successful World TB Day event and success in your continued fight against tuberculosis. Thank you.